Hi, I'm Paul Subchik,
And I'm Jeff Minlin.
and this is the cytoskeleton, which consists of microfilaments, intermediate filaments, and microtubules.
So first we're going to talk about microfilaments, which are made of actin, so they're also known as actin filaments. This is a fluorescent stain of the actin filaments. Microfilaments are usually around 7 nanometers in diameter, and as you can see here, they're usually pretty long. Microfilaments are made of actin monomers. One microfilament strand is made of 2 actin strands that are intertwined. They are located right under the plasma membrane, which is right here. These are the microfilaments. What they do is they resist tension, which is the pulling of the cell membrane. Microfilaments are mixed with myosin strands. Myosin strands move along the microfilaments to contract and relax muscles. So on muscle cells, this happens on a very large scale, which results in muscle contraction. For a lot of other cells, it helps cell movement, contraction, and cytokinesis as well. In amoebas, the microfilaments squeeze the cytoplasm in a direction to move. So in this case, the microfilaments would squeeze the cytoplasm in this direction. This area is called the pseudopodium, and that causes amoeba movement. Some white blood cells also move like this. In plant cells, they make a sort of racetrack for the cytoplasm to circulate along. So that's called cytoplasmic streaming, and it helps distribution of materials in the cell. Next we're going to talk about intermediate filaments. Here's a picture of intermediate filaments. They are actually out of the 3 types of cytoskeleton filaments. They're most enduring, especially after like chemical treatment. They'll still be there. They're in like dead skin cells and stuff, so there's usually a layer of these on your skin. So here's the structure. Intermediate filaments are made of these monomers of various proteins that intertwine to become dimers and then tetramers. Sheets of tetramers then bundle up into what's called a supercoiled sheet and makes this filament bundle. They're usually around 8 to 12 nanometers in diameter. One of the proteins that's commonly found in intermediate filaments is keratin, which is found in your skin and your hair. So this is a rat epithelial cell. As we can see here, the intermediate filament network holds organelles in place, like the nucleus here. It also lines the interior of the nuclear envelope, and it is in extensions of nerve cells. It supports the transfer of like information.
Lastly, we'll talk about microtubules. They're made of tubulins, so they're also called tubulin polymers. This is a fluorescent stain of microtubules. During mitosis, microtubules are 25 nanometers in diameter, and the structural function of microtubules is to resist tension. Microtubules are made of alpha and beta tubulin dimers, which form polymers of tubulin polypeptides. The walls of microtubules are formed by 13 columns of these tubulin polymers. Cilia and flagella are special arrangements of microtubules that extend out of the plasma membrane, and they aid in cell movement. They're formed by a ring of 9 doublets of microtubules with a pair in the middle, and each of these doublets is held together by a protein called denine. Denine walking refers to how these cilia and flagella help the cell move. The denene of one doublet grips another doublet and pushes it either up or down, which causes large-scale movement of the entire cilium or flagellum. Microtubules originate in the region near the nucleus called the centrosome, and the centrosome usually contains 2 centrioles arranged diagonally, which consist of 9 triplets of microtubules.
Okay, so as we can see here, the cytoskeleton consists of like intertwined filaments, microfilaments, intermediate filaments, microtubules that work together. Without them, the cell would lose its structure, its ability to resist tension and compression. It would lose its ability to move, things like that. A lot of cell circulation would have a lot of trouble. So, like vesicles, for example, also rely on microtubules for movement, where they latch onto the microtubule and move around the cell like that, distributing materials. So thank you very much for listening to our presentation.